everyone. Today on Genomify, we're covering something that I've been working on for a while now, and that's an RNA-seq app I built from scratch that takes you from raw data to publication-ready plots in under 2 minutes per sample, and you don't need to write a single line of code. All right, let's get into it. All right, so this is the RNA-seq app I've been developing for a couple months now. It is a graphical user interface that you essentially just click around, select your raw data, tell it what reference file you want to use, and then run the pipeline, and I'll show you all the analysis steps later down the line. But I was just going to walk you through a little bit of how this app works, and I will attach a download file and another video I'll show you how to actually install it. But this will just be a quick run through of how the actual application works. So you first start off with the select data tab, and essentially all you're going to do is navigate to your Finder, and you're going to click a folder on your local computer, it can't be in the cloud, that has your raw data in it. In this case, it's just going to be fastq files, and these are paired and fastq files for this example. And then you're going to hit scan directory, and then it's going to automatically detect the read 1 and read 2 for each sample and show the number of samples in each directory. So that's kind of the first step. Just kind of walking through generally more here on the left. It'll show you your available indices, which I'll show you in just a second. It'll automatically detect your CPUs you have available on your computer. I recommend bumping this up to as high as you can go to make it faster. And I've attached these kind of methods as well as instructions to uninstall if you're having trouble or just no longer want to use this app. And also recommend using this quit app button just to make sure Streamlit actually cancels when you close the tab. But yeah, so once you've loaded in your fastq files, you'll then go on to make your reference index. Okay, so now that you've loaded in your files, you're going to click this reference index tab. And I've set this up in a way where I have 4 preloaded reference files, 2 human, which are just different versions, and 2 mouse, which are just different versions. And if you select this, it will automatically download the reference transcript FASTA file and generate the Salmon index. If you want to use your own reference file, I've integrated this build from FASTA file. And in this case, you'll just, same as before, select your, the transcripts .fa.gz file. You'd select that, and you'd hit build index. But I'm not going to do that today. I'm actually just going to pick the preloaded human GenCode version 36 index, and we're just going to hit build index. And this is going to take around 3 to 5, maybe 10 minutes, depending on how many CPUs you actually have available on your computer. But I'm just going to let this run, and I will hop back on when it's done. All right, so it just finished running, took a little over 5 minutes, and when it successfully has created your index, you'll get this green box saying index already downloaded, and you'll see your index appear here in this top left. And the beauty of this app is once you've done this once, you actually won't have to do it again. It'll save these indices in a folder, and it will automatically detect which ones you've already created. And in the future, if you just wanted to select one that you've already run, in this case we only have one available, but you can have multiple references, you can just hit select and move on with the actual analysis pipeline. And so once you've selected your files and created your index, all you have to do is run the pipeline. And so here it's just showing you that you have your index and your samples ready. So the 2 green boxes means ready to go. In this case, I will say you can rename your run to whatever you want. I'm just going to call this demo test. And each run it will save to a different directory. And so you can always refer back to your runs after you've done this, even after you close the app. And I've also put in these pre-processing options. They add quite a bit of computational time as they're computationally intensive. The first is this fastq, which is a pretty standard technique bioinformaticians use just to get a sense of what your sequencing run looked like. I highly encourage you to run these just to get an idea if your sequencing run actually was successful. It actually will not affect the analysis whatsoever. It just was going to output a fastq HTML for each sample. I've also added this trim galore option. For RNA-seq, especially for publications, it's traditional that you trim the adapters for the actual reads, but it actually adds quite a bit of time, and I've found that it actually doesn't change the analysis too much. So I'm going to leave these unchecked. They're totally not necessary, but I've added them here just in case you want to use them. So as you can see, it plops out this estimated processing time, and if you select these, it goes up quite substantially to about an hour. But if you don't have them, it's only going to be about 6 minutes for 4 samples, and if you change your CPUs, it will update that time estimate accordingly. So pretty much all you're going to do to perform your RNA-seq quantification is hit start quantification. And this will kind of give a real-time output of which sample it's on, as well as the estimated remaining time. And yeah, so I'm gonna let this run and I will hop back on when it's done. All right, so that took a little bit under 8 minutes and everything completed successfully. If there are any errors and your sequencing data is strange, it will kick back an error and output what the error actually is, and you can troubleshoot it that way. But everything I've tested has run successfully. So please let me know in the comments if you're having issues with this analysis pipeline. I'm happy to help you troubleshoot it. So once that's finished, you'll then come to the results tab. And so as you can see, I have a couple different runs here, but I'm going to show you the current session here. And that is going to show you very high level, just QC stats for your samples. And so it's going to output the percentage of reads that were actually mapped to your reference genome. I like to have samples that are over 80%. That's typically a good measure that your sampling or your sequencing run was successful. And it just outputs this in a graphical interface. And it also just gives you a counts matrix and shows you the most highly expressed transcripts. These aren't super useful for publications. This is more just for you as a user to know if your sequencing run was successful or not. So for the actual analysis tab next, this is going to be where the really fun starts. This is the stuff that took me forever to do before. You had to go into R Markdowns, change all the code manually to get all your code running. It was just kind of a pain in the butt generally. So I just wanted to automate this, and I just wanted to share it with you all. It's using the same underlying code as all the other videos that I've made for this channel, just making it easier to use. And if you are curious what code is actually being used, I've included this analysis code drop down. It's essentially what's being used to actually analyze the data, but you don't really need to worry about that. Unless you're curious, please take a look if you want. So what you're going to need to do next is add the metadata. So I've kind of created this interactive, clickable table format that will allow you to add whatever you want. I'm going to just put in my comparison I'm testing here. You can add replicates, you can add additional columns if you want. I'm not going to do that here, but if you want, please feel free to do that. And you have to just hit save metadata so that the application is going to recognize which sample belongs to which condition. You can also select which condition or metadata column you want to color your PCA by. So the first thing we're going to be able to do is run a quick PCA analysis. And these are going to take maybe 5 to 10 seconds to run, but it's going to output a publication ready plot. And it's going to show you additionally how much each principal component is contributing to sample separation. And you're going to be able to download all of these files for all the analysis steps, as well as the raw data that is being inputted into the actual analysis, or outputted by the analysis, not inputted. So then next, once you've convinced yourself that your samples are clustering like they should, this is a very good upfront QC step I use in my sequencing analysis to make sure my samples are doing what I expect. Next, we're going to perform differential expression analysis. And so I've made this super customizable. You can change your comparisons to whatever you want. If there's more comparisons you want to make, it's super easy to just change. Here we only have 2 options, but if you have more samples, feel free to go crazy with this. You can also change your log2 fold change threshold, as well as your p-value for the ones you want to call significant. So all you're going to do is just hit run differential expression, and under the hood it's going to be running this DE-seq2 analysis like I had shown in the previous RNA-seq tutorial video, but here you just have to click a button. And so it's going to output this interactive plot, so you can actually hover over the volcano plot to see which genes are actually significantly differentially expressed. You can also type in genes to actually highlight where a gene is on the volcano plot, which is a really cool thing. And you can also label the genes using this, and also you can drop down just to see which genes you want to label here. But also there is a more publication-friendly TIFF file that you can download here. And again, you can download everything you want here, as well as all the outputs from DE-seq2. If you want to go back and plot it yourself, please feel free. This is more just a quick and dirty tool to look at the data very quickly. And this is also just going to be kind of a quick output to see which genes are actually being significantly expressed between the two conditions. And then one thing that I really did not like doing was looking at gene sets in volcano plots. It's a really helpful technique, but hold on, let me actually come back. So gene sets, if you've seen my video, is essentially just a list of genes that are associated with certain pathways. And I like to overlay these on volcano plots to see if there's any trends for a particular type of gene set. And so I provided the link for a MSigDB website. And in this case, you can download the hallmark or whatever gene set you want. But what you're going to do is just hit this gene symbols. I'm going to look at hallmarks first, but you can look at any of these gene sets you want. And then we'll come back to the RNA-seq processor, and you can come back and select your actual gene set file here, which is the GMT file. It's going to automatically recognize all the different pathways that are in this gene set file. And so I'm going to look at the EMT transition because I know that one is super important in this particular comparison. And it's actually going to highlight all the genes in that gene set on the volcano plot. And you can interactively look at it just like before. Or if you want to look at the TIFF file that you can put in a publication, it's right here. This is exactly the same plot I put into my paper in Nature Communications. So really there's not much you have to do. And it's really easy to iterate over different pathways. You can just click another one, hit highlight, give it 5-10 seconds, and out pops the other gene set mapped on top of the same volcano plot. And this one doesn't have as many genes, so you're not seeing any that are labeled. But yeah, that's a really cool technique. This took me forever when I was doing it by hand, and so this just really speeds up the different pathways you can look at when performing RNA-seq analysis. And then lastly, if you're unsure of which gene set to actually overlay, like for instance, you have no genes in the E2F but tons in the E2 epithelial to mesenchymal transition, you can actually look at all the gene sets that are in this GMT file that you've loaded and see which ones are coming up as significant. And this will help you hone in on which of the pathways in Hallmark or whatever gene sets you're looking at to focus on in your volcano plot. So in this case, I already knew EMT was going to be a top hit just because I've looked at this data before, but you could look at these other gene sets, see which genes are being upregulated and downregulated, and really just help you drive future biological questions that you want to ask from this RNA-seq data. And again, you can download all the images as well as the raw data that's going into this plot. So that's pretty much it for this application. I've managed to distill what used to take me a couple hours to something I can do in really under 10 minutes. And I really hope you guys are going to be able to get some use out of this. This is the first time I'm releasing it to the general public, so please let me know in the comments if you're having any issues. And after this, I'm just going to show you really quickly where you can find this and download it to your computer. And I'm also going to create a new video on how to actually install this after I upload this. So thank you. Please subscribe, like, comment, and let me know how you like this application. Thanks. Just so really quick, just to show you where you can actually obtain this application. Here's my personal website. I put a lot of my my work as well as what I do on this website, but I've created this link that will allow you to automatically download the DMG file, and that will automatically download to your computer. You can also go to the GitHub page here and actually look at the various different releases I have. I'd recommend version 1.1, but I will be continually updating this as people come back with comments or problems. But if we come back here, I'll just really quickly show you how the actual DMG file works. So what you're going to do is you're going to click this DMG file and you're just going to drag this into the applications folder. I actually already have it installed. I'll do this more in depth in another video, but essentially all you're going to do is just drag that application into the applications folder, give the permissions for the Mac to actually use this because it's not a credible developer, but trust me, it's not malware. And we'll get more into the installation in later videos, but I just wanted to quickly share that in case you wanted to just do it quickly. But yeah, thank you everyone and have a good day.